Hi, everyone.
Hi, good morning.
Good morning. How is it going?
Um, it's it's a slow morning, trying to get a lot of stuff done. So it's
How's it going with this class? Do you have questions specific questions with this topic that we have right now, the muscle physiology stuff?
Not at the moment, just trying to get through the assignments first, and then since it's still the beginning of the week. So
No, I meant from last week's.
last week.
Yeah, the muscle physio, I think.
Yeah.
We're gonna spend some time with, or so. Those things are good for you, understanding wise.
I went over it a little bit, but I haven't exactly completely went through it yet. So
okay, that's okay. Then we'll go through some of that today, so that might help too.
Okay.
Hopefully, make it a little easier to get through those things.
Mm-hmm.
How's everybody else doing? Good morning. I'm starting to have a few people coming in here. Well, we'll wait a little bit longer then. Let me just put the recording on mute for a moment. Well, I think one thing that let's just start, since people are probably going to add on, and people, are you guys not sharing because did you guys not do the muscle physio yet, or you don't have questions on that, or did you guys work on that yet? Anybody, if you could help me out with that a little bit. Hello, am I not hearing you guys, man?
Well, I wasn't able to really go over it or do it. Like, I remember some from previous science classes, but really about it. So
okay,
so I haven't gotten to it because I'm kind of behind on my assignments. So.
Yeah, no, that's totally fine. That's totally fine. Hi, is this Nina there?
Good morning.
Good morning. I know you got questions. I'm not sure the participants so far have too many questions of the material or have done it. So let's get started with what you got going on with the muscle physio, right? That's where we want to catch up.
Yeah, so basically, if you go down to slide 5 onward, that's where I'm just a little bit confused. I get a general idea of the sliding filament theory, but not quite.
Okay.
And I'm wondering, what's that?
Can you see my slides?
Yes, I can. And I'm seeing if you go down to slide 5 to start,
okay,
that's kind of where it starts.
Got it. Yeah, then let's go through that a little. Maybe you and I can start a little dialogue, and then people chime in. I think some people are just getting to the material. So you guys, if you have questions when it comes in, just ask the questions, okay? So
I did want to bring up one other thing, Doc, and that is that I realized last night that the lab I should have started last week because it was doing something over 7 days. If we turn it in next week, can we get all the points? Because it's doing the exercises.
Which hold on, let's go to that real quick first. We'll fix that real quick. Let me share that with you guys.
I just figured I'd at least tell everybody in case they haven't gotten to the lab that.
Yeah, no. I want to make sure we'll do this stuff right. And and most likely I made a mistake somewhere.
That's okay. I mean, if you go down to lab.
This week's lab or next week's?
Yeah,
lab?
This week's, the posture evaluation.
So
that that would have required us to start last Tuesday, and I looked at it last night, so it's not ready to be turned in yet.
Got it. We'll do that a week later. That's a good point. I didn't even realize that. Right. Yeah, all these
still about this stuff. I figured I could just bring it up.
Oh, please, please. There's
Yeah.
so many details, and I'm like ADD, so I'm trying to get it all together. I might be okay making a class lecture, but the details are a little hard sometimes. So I need all the help all the time. Good. So that's that, right? So that's good with that part. So then let's go back to the lecture. What am I doing here? Trunk picture.
So part of what I was trying to understand is that I was trying to, like, in my mind, relate the ATP breakdown and recreation back to what we've already learned. So to kind of solidify it long term, but I still don't quite understand. I still don't quite understand it. I think one of my first questions is I understand there's creatine phosphate, that's where you get that additional P for the adenosine triphosphate, but where does the creatine phosphate come from? Is there an organelle for that?
No, that's floating around. That's brought in.
Okay, so it's kind of its own organelle?
No, it's a molecule.
Like kind of like the calcium? Is
like
it?
It's
is
like
it
a molecule like
charged?
ATP itself as well.
Okay.
So
Okay.
it's the creatine is a part, and then the phosphate is a part, and the phosphate splits up and goes back to the ATP, the ADP to make ATP.
Right. And that, so that was one of the parts I didn't quite understand, and I was trying to solidify it. And then,
yeah, we can go through that. Yeah, we can clarify that a little bit more. Absolutely. Is that what? Let me start with the beginning of the chapter, since some of you guys haven't really looked at it that much. So when we look at here the physiology, we're really looking at muscle function. How does the muscle actually do its work? And when we look at what is the muscle's work, we're basically looking at it. It's trying to move. It's trying to shorten. It's trying to move bones around, or it's trying to hold something in position, or squeeze something. If you're looking at the gut tube, like the mouth, that's a squeezing muscle. We talked about that when we did the facial muscles. But basically it accomplishes that by shortening, and shortening meaning it comes together, the ends come closer together. And so what we're trying to do in this chapter for the most part is understanding how does it do that shortening. And to back up a little bit, and of course, when it does that shortening, it does all that stuff, all these functions. You can go through that in the video. The first beginning is there also some characteristics that are there shortening. The shortening is this contractility. That is that active portion that we're going to work on. But also muscle tissue has to be able to be irritated by the brain or something else, but a lot of it is by the brain, that it needs to be responsive, that if the brain says we need to use that muscle right now, it's got to respond to that message that the brain sends. So that's partly what we're discussing here, responsiveness. These are all terms that are meaning the same things. That's why I put them together. And then the muscle needs to be passively, again, extensible. If I shorten it, I need to bring it back to be its original length, or even a little bit longer potentially. It's extensibility and also elasticity. That's actually a little bit longer. And so those are all passive parts that the muscle has, passive features, but this contractility is the active part that we really want to understand how the muscle functions. You're looking at muscle terminology. You go through that in the video. That's easy stuff. That's prefixes and suffixes. All these myo, mesio, sarca also refers to muscle. So when you see those prefixes on a word, you know it's referring to muscle somehow, and you can at least go down that path in your decision tree. What's going on with this sentence? Sometimes it's a foreign language, right? And then the next thing here is the type of muscles. We have some muscles that are the ones that we can think about, that contraction of an elbow. I need to contract this biceps here and make it shorter. That's a skeletal muscle. That is the main type that we really study. But we also have two other types. We have a heart muscle that's specifically for the heart. The specifics there is the heart needs to contract as one unit, even though it's many, many different cells. And so we have these features, these channels that send the muscle impulse, the nerve impulse through the whole muscle at sort of one moment that makes that whole muscle contract as a unit, or in synchronicity, because it's actually a wave that goes over the muscle. So the top part of the heart contracts a little bit fast first, and then the bottom part of the heart contracts. And so we'll talk about that, but that's a specific type of muscle. We only find that in the heart. But you can see that these intercalated discs here, if you go back to, starting it further, those are those little channels that let the electrical impulse go in and out, these gap junctions. And then we also have a smooth muscle, and that's the muscle that's around the gut tube, for example, or the blood vessels as well. Wherever we have tubes, we have some muscle around that is smooth muscle. It's a little bit different. It's smaller cells. It contracts rhythmically. It doesn't function. It functions the same, in fact, that it contracts. So those parts are the same, but then the uniqueness is how does it do that, and how long can it keep contraction and things like that. But what we really talk about in this chapter is the function, is the skeletal muscle. And so this is where we pick it up, Nina, right? This
yes,
is where
please.
Right, 5, right? So so you stop me, I make the story, and then you fill it in if you could, as much as possible, or everybody else too, not just you, of course. But when we then look at this muscle, skeletal muscle, we wanna look at a few specific organelles. We talked in the cell structure about organelles, about the cell membrane, et cetera, et cetera. And so here we have all those organelles, or many of them, but the specific ones that we want to look at in terms of understanding how muscle works is the sarcolemma, and that's the muscle membrane, the cell membrane around the muscle. And that is the uniqueness of that structure is it can transmit an electrical impulse. And so when a nerve fires down an impulse and says, hey, muscle, we need to contract because we need to go walk to the door, and you need to start making these legs move, that's muscle contraction, that movement. The nerve impulse goes to that sarcolemma and spreads over the whole muscle as a message, as like a shock wave that, yes, now we contract. So that's that message. It's an on and off message. Nerve impulses are on and off sort of impulses. Where the variety of nerve impulses happens is when we talk about neurotransmitter. But in muscle, it's always the same. It's always that activation one. And then the second term here is myofibril. So when we look at a whole muscle cell, a whole muscle, as a whole muscle, like from the knee to the hip, that quad muscle, that is those are many, many long muscle cells parallel to one another. So they're interesting. They're multinucleated, they call them. That means they have more than one nucleus because they're so long that in the embryonic stage, they have to single cells have to fuse together and make one long one. And so the nuclei that those individual muscle cells had stayed behind. And so that's why it's called, muscle cells are called multinucleated. And so then this whole thing here is one muscle cell. The sarcolemma is around the muscle. And so that's one thread that goes, let's say, from the knee to the hip, the whole distance. And inside that muscle thread, we have small little tiny muscle fiber looking things, and we call those the myofibrils. And so those are like, instead of muscle fiber, I'm thinking of like fibril is a small little tiny muscle fiber. That's how I remember the terminology. So the muscle fibril is an organelle, really. That's the shortening, contracting organelle that we're going to understand how a muscle functions on its microscopic plane level. But the muscle cell itself is this big structure. Then inside we have these rods, and these are these contractile organelles that are composed of, if we go to slide 7, if we're looking at slides over here, they are composed mainly of the contractile or proteins that we need to make that muscle shorten. And we have 2 of them. We have one that's called the thick filament, also known as a myosin, and one that's called the thin filament, also known as the actin. And the actin is kind of like a thread that sort of lays there. And if you look, Yeah, it is here. If you blow this up a little bit, maybe I could blow this up a little bit. If you blow this up a little bit, you see how the thin filament here, threads go towards parallel. This is a myofibril. This is a part of a myofibril. I should actually maybe say that first. This is part of the myofibril. It's called a sarcomere. So if I take that myofibril here, that one thread organelle, and look deeper into it, then I get to this picture here. And so this here is that myofibril, but now we're only looking at one section of the myofibril. And they call that a sarcomere. Again, sarco is that terminology for muscle. And so actually the way I remembered that was it's merely a muscle. It's so tiny. It's not quite a muscle. Sarcomere, merely a muscle. And so that's how I remembered that gradation. So it goes from muscle cell to myofibril, now to sarcomere. So we're looking at the smallest sort of level that we want to look at and understand how that thing works. And so then when we analyze that, that's when we get that thin filament is one of the thread types, and the other, and that's called actin. Here, actin. And then the thick filament is the other type. That's the thick filament. That's the myosin. Here's a myosin. The myosin has a head and a tail. The myosin has a head and a tail. The myosin head is really what does the work. That's where we're going to need the ATP. See, you see the dotted line, and then the picture part. That motion is what does the work. That myosin head can move.
So that turns from ADP to ATP, or ATP to ADP?
ATP to ADP.
Okay, so it loses a phosphate.
It loses a phosphate when it does that work.
Okay.
There's one more step. We'll get to that in a second. Yeah, but that's exactly there. And that's the ATP to ADP. Whenever your body does work and it needs energy, it takes that ATP molecule and chops up a phosphate, and that phosphate gives a lot of energy. It's like a spark. You think of like the ATP to the ADP, that split off of the phosphate. It's like a spark. That's that energy part. And so that's what is necessary to move that head around. All right. Then so that head attaches to that other thread, or actually it attaches and it deattaches. And that other thread is that thin filament, that actin filament right here. And so when we look at that actin a little bit deeper, how do I move that? Okay, when we look at that actin a little bit deeper, we're going to have, what's most important here is we have these beads, and on those beads, we have an active site. And the active site is where the myosin head can attach to. Think of it as a magnet. It's a magnet between the active side of the actin and the head of the myosin. And then the thing is with this, and so what happens when you actually then have a muscle contraction is that actin, the myosin head attaches to the active side of the actin and moves that head this way. And when it moves it this way, the myosin slightly pulls on the actin this way, and they shorten a little bit. The ends here come a little bit closer towards the midline. See this M line here? There's a lot of terminology to it. I try to keep it sort of to a minimum. They also have these terms here. This is the M line and the midline, and that's where we anchor the myosin heads or the myosin, and then they go out each side, and then they have these actin threads that come from that C line because it's zigzag looking, and that's where they're anchored. And then moving those heads slowly pulls the Z line closer to that M line, and that slowly shortens that muscle, brings it together. Does that make sense in concept? Uh-oh, did I lose everybody?
Yeah. Ish.
Yeah. Ish. Well, we're going to do it a couple of times. But that's sort of the essence of what's really going to happen. So, right, we got that long muscle cell that spans the whole muscle. Inside, we got these long fibrils that have these Inside, they have all these actin and myosins that look, attached to one another. And all of those, the whole length, all those little tiny heads hook up to these, from here, these myosin heads hook up to this actin, this other thread, and brings these two threads closer together, and over a whole span of a muscle, that shortens a muscle. You think like this is 10 times, gazillion times, and then a whole muscle can shorten. That's only like one place. But we want to study that one motion in that one place so we understand it on a microscopic, actually on a super microscopic level. When we fill in a little bit more, one of the things we will have to understand here, when this active site is empty, is free, this myosin head, when there is ATP present, automatically this magnet will connect. The myosin head wants to attach to the active site and shorten and move that towards the midline.
Okay.
Automatically when there's ATP present. All right. And so what we need to do is we need to have a situation where we can say like, well, we don't want to always contract that muscle. What we got to do when we don't want to contract the muscle, but we want it to be ready to contract. So we want ATP present. We want all this situation ready to go. So what we do then is we say, well, we could cover the active site. So now we've got this other thready molecule, this brown, grayish one called tropomyosin. Don't have to worry about the name, worry about the concept, okay? This one covers the active site. And then you got a third one, and that third one is this troponin here called, and that moves that tropomyosin over the active site back and forth. And when it's covered the active site, the muscle is relaxed. When it moves away from the active site, the muscle will contract automatically. So that when ATP is present. So now the control is not really the ATP down here. The control is the active site open or closed, or covered or uncovered, right? So now then that becomes the trigger if the muscle is contracting or not. So the ATP machinery always goes. So let's talk about the ATP machinery a little bit more, because that's that sliding filament theory. So when we look at this, so here we got these red ones are the myosin heads. These are the myosins. This is the thick filament, and this is that thin filament. So when we look at that cycle, basically what we're worried about is a few things. We're concerned about the myosin head attaching to the active site, and then we're worried about the myosin head moving and then detaching from the active site and getting back into a position where it can reattach. So what happens is that the key here is the ATP actually does not do the actual movement. What the ATP does, it takes the myosin head from a low energy position where it's already moved and it's finished its movement. Here it finished its movement, and it brings it back to the high energy position where it sticks straight up, and then it can reattach passively. I mean, it's not passive, but it's without any more energy input. It can reattach to the active side and does this movement automatically, and then the ATP comes back in and it cocks that head back up. So it's like a spring. So the ATP brings the spring into a position where it then can just be let go, and that's then the motion.
So I kind of want to so obviously what's happening is the muscles contracting or shortening.
Right.
So so that means, like, say we're using our bicep. I know we use that for a lot of examples.
Absolutely.
So I see on the second one, which way is the head sliding if I'm contracting like this?
Well, the head is going from here
towards the site,
from
towards the point of origin, or towards the point of insertion?
Well, no, that one is actually that doesn't matter, because you got heads going both directions.
Oh, okay. I thought maybe it was pulling them shorter. That's what was happening.
Huh?
I thought maybe because it's like contracting and pulling them shorter, they were all facing one way.
Yeah, no. Well, that would be good, but the body is even better. because the body
Okay.
says, well, I can have the actins taken from both sides.
Okay.
And then I can have the myosins from both sides pulling on it. And instead of just one side shortening, both sides will shorten. That will double my shortening.
Got it. Okay.
Right? So that's why the heads go both directions. And the other thing is the muscle shortens, yeah, when the head goes when the head closes down, right? And it shortens, it cocks, or it uncocks, actually, it releases.
So
head.
it almost,
That's
it
when
doesn't
it's
matter
short.
the directions, just that it is,
Oh.
it is moving.
That's what it's moving. And the other thing is you got so many heads. When you look at, is there, do I have a picture of that? There is somewhere, there's pictures where you can see, you got so many of these things going on. They don't always do it at the same time. There's like a tug of war. Some people are pulling on one side, the others have to grab the rope again and pull, and the other ones are pulling. It's the same thing. You got some are working, the other ones are recocking, the other ones are working. And so it's like a machine. They don't all do that at the same time. And that also makes then, of course, the muscle shorten more because one little pull goes a little bit, a little bit, a little bit, a little bit. And so they all have to kind of work together. That whole thing, like, you know, that you think of a massive arm, you know, for these small little critters, this arm is like a friggin' 3-ton machine thing. And they all pull a little bit, a little bit, a little bit, a little bit. And so that's that mechanism of how they do that. Does that get a little more sensible? All right. Because now what we got to do is we got to understand. So this is when there is ATP present and the active side is free, this is automatic, not passive, automatic. It just happens. So now we got to figure out how can we control it happening from not happening. And so now we got to talk about how we're going to cover the active side and uncover the active side. All right. So I'm doing it a little backwards, but that way we can get the story. So when I look at the active side and the inactive side, then we get calcium into the mix. So when calcium is present in the system, when it's released, it moves this thread that covers the active side away from the active side. That's all you have to understand about this slide. When calcium is present, the thread is moved away from the active side is free, the muscle will contract because the head will attach to it. Again, make sure ATP is present. But if ATP is not present, you're dead. So it doesn't really, it's a given that ATP is present. Well, I shouldn't make that personal. That's his people. And so when calcium is present, then the active side is free, the muscle can contract. When calcium is not present, the active side is covered, the muscle cannot contract. The head cannot attach to it. And so now what's important here is how do we make that calcium? How do we work with that calcium? And that's where we get into the slide number 6. And so when we have a nerve impulse, we got to spread that nerve impulse over that sarcolemma, over that cell membrane, and then that nerve impulse actually penetrates through these yellow lines in here around every myofibril. These are myofibrils. And see, this is actually a picture where you can see all these red dots are all the myosins sticking in and out. And all these white things around it are all the actins. And so the myosins have head all around them. And all around them, they attach and pull on that actin. So you got gazillions going on of that thing. But anyway, that nerve impulse comes down through the sarcolemma. It penetrates deep into the muscle going down that yellow line. And that's nerve impulse, that yellow line. And then it activates that blue bag. And in that blue bag, we have calcium. So this is a reservoir for calcium, this blue bag. And it's known as the sarcoplasmic reticulum. See here, sarcoplasmic reticulum. And it's like an endoplasmic reticulum in every other cell, but it's completely different. It's not like making things. It just has that same fundamental structure. So it's very different function in the muscle cell. But the name comes from that part. So the sarcoplasmic reticulum is actually a bag full of calcium. And when a nerve impulse comes down that yellow thing, it activates the SR, sarcoplasmic reticulum, to release all its calcium into the myofibril, and then that whole thing can happen. When the nerve impulse stops coming down these two, all the calcium will be sucked back into the sarcoplasmic reticulum, and then that whole muscle attaching to the active side can happen because the active side is covered with a thread. Do you understand that?
Well, that's what you talked about with rigor mortis, is that like if it's flooded with calcium and the sites are opened up, but there's no ATP to fill it, nothing happens. It's just super stiff because it's charged with the calcium in there, like ready for action, but there's nothing to activate it. So it's just kind of stuck.
It stays in this attached, it stays in this format. I think it's this one. It doesn't let go. Right. And obviously the disruption is the calcium. If you're dead, rigor mortis happens after a while after we die. And so if somebody's dead, the calcium, the integrity gets lost. So the calcium flushes into the muscles. And then all the active sites are open. And the rest, the ATP we have, is used up. And once it's used up, that muscle active side open, the myosin head's attached. It doesn't let go. And that's that stiffening of the muscle.
May I ask another clarifying question?
Oh, absolutely.
So you talk about, we have the isometric and we have isotonic contractions. Is this occurring for both isometric and isotonics?
Yeah. Let me open it. Let's take that as a number 2. Let me open it to the other people. Did you guys get some feedback for a second? Then Nina, we'll go back to that. Did you guys understand what I was understand what I was talking about so far? Can I get some feedback from the other people in the class?
Yeah, it was I understand it.
Okay. I mean, it might be terminology, might be a lot, but you know that you good. Are there any questions that you guys have to that? Any points I should still clarify on that, or you guys are pretty good at this moment?
I feel good about it. This makes a lot more sense.
Okay, and then this is in the video too. This is a very good connection slide where like this is the nerve coming down, nerve impulse going in. This is the SR, the calcium going in, and then this happening. So this is a good slide to sort of review all of that in one's mind. And once you understand this slide, you're pretty good. All right, good. Then let's go to that thing. You want to talk about isometrics and isotonics, right? Is that the thing, or the
well, I don't have, I understand it, but I guess what I'm wondering is because I thought they had to move in one direction, and they were shortening, I didn't know if it was the same process occurring when you're doing like an isometric, like when you're holding a plank, for example, versus when you're bending over and flex.
Right.
Inflection.
Yeah. It's totally the same.
Okay, that makes sense now that you've explained the heads can go different directions.
Yeah, so the concept of that muscle contraction, how that sliding filament thing works, that's always going to be the same now for every muscle in your head, for every contraction. It's all the same on that little level. And then when we extrapolate, when we go to isometric versus isotonic, it's just in the isometric. Iso means same, metric refers to meter. That's a distance. So in an isometric contraction, you don't shorten the muscle, you hold something. But on a microscopic level, that's still that muscle. The heads have to attach to the actin and hold it in place. They might not move it, but they kind of move it because they all have to move a little bit so it doesn't slide backwards. Right? So they have to do the same work. And so an isometric contraction is you don't move the muscle, you hold like a grocery bag coming into the house or something. That's isometric. When you have an injury, when it goes into that discussion, then isometric is the first way you want to kind of rehab a situation, because you don't have muscle movement. I mean, you don't have joint movement. So let's say you injure your back. one of the first things you want to do to rehab is you want to mobilize it passively and do a few things like that. But in your rehab, you want to do holding patterns, holding muscles, so you can strengthen without moving joints that may be hurt. And so that's where isometric comes in, like a lot of the time when we come to rehab. And then isotonic is just the fact that the muscle moves its distance. So the tone, the iso, the same refers to the tone. So when I move a bottle up, the muscle tone, how I contract the muscle sort of stays the same, but the bottle moves, so have motion in it. So that's what the reference is in terms of the word. But for us, it's just movement. And then we go with the movement. We go and differentiate between concentric, a shortening contraction, and eccentric, a lengthening contraction. So if I let go of a muscle, that's still an isotonic contraction, because the muscle has to slowly let go. Otherwise, it's just boing. Then it ain't contracting. I can't hear you, Nina.
But there's no energy loss when you're releasing. I mean,
there's a lot of energy loss.
There is energy loss.
Yeah. As a matter of fact, the eccentric contraction is the best muscle builder, generally speaking. So when you actually do exercise, when you do like curls, you don't want to do like, you want to wear out that thing back there, the elbow. You want to let go of that weight slowly, because that eccentric contraction is actually where you build a lot of muscle. That makes sense? I mean, maybe not that you build muscle.
It does, like from a gym perspective, but I'm just trying to figure it out from like the science. Like what's happening.
Yeah, again, you're thinking of a degree of how many heads move at the same time.
Mm-hmm.
So when none of the heads attached to the active side, the muscle just flattens out.
Okay.
Right? Let's say you can hold passively shorten, that's fine. Muscle actually has that in it, that it actually does shorten with all those myofibrils, all those things shorten. And then I slowly lengthen it with resistance. So that's the thing. I slowly lengthen it with resistance. That's the eccentric contraction, right? But in order for the muscle not just to go limp all of a sudden, the myosin heads have to attach to the actin and slowly attack a little less, move back a little more. But it's a gradual process.
So if I'm holding a curl like this, and I'm not moving, but there's some resistance here, the sliding filaments is still happening in the muscle because there is contraction happening. But when it's here, nothing's happening because the muscle's not contracted.
Well, only the muscle is only the muscle is completely limp. I use this as the muscle is completely limp.
Okay, that makes a lot more sense.
So it's like the sliding, it's almost like the sliding filament theory pretty much always happens. It's just to what degree.
Okay.
It's like you can look at it like it's a volume knob going up and down. Like if you want to contract it, you're going to pull it up a little bit, more myosin heads. If I want to lengthen it, a little less contract. And I bring the volume up and down, and the volume meaning how many altogether heads contract at the same time.
Well, and if you think about it, that makes sense, because we have, the muscles that perform movements, but then you have your posture muscles that you're talking about the degree. So the posture muscles, it's probably a smaller degree because it's just holding you up. But if you're like heads forward and you're fighting gravity, there's more work has to be done to fight the gravity.
And you just very well explained this slide.
Okay.
So we have two different muscle types. So we have multiple, not just two, but the two we can sort of differentiate. We have the slow twitch and the fast twitch. So the ones you described as the postural is the slow twitch. So it doesn't react like a quad. It can't kick a ball that fast. Right? But it holds the thing. And so it has a different composition a little bit. It has a lot more myoglobins. And myoglobins are like hemoglobins. They carry oxygen to the muscle. Oxygen, we need to make ATP. So you always, oxygen, ATP, you always connect those two together. That's why we need oxygen, to make ATP. And so myoglobins are a red thing. So these slow twitch fiber, they're looking general a little more reddish, and they have more of that hemoglobin magnet kind of molecule, which is called myoglobin. And so that attracts oxygen, and then that oxygen can be used to make ATP. And in a postural muscle, we use a lot more ATP made that way to continue that contraction, a little slower contraction, but a little less forceful, but longer lasting.
And so, like, if you're running a sprint versus running like a mile at a steady state, if you're not oxygenating your muscles fast enough, that's when you get fatigue. And that's why you can run a sprint, but you can only run that fast for so long.
Yeah, and that's where I don't exactly know how the distribution is. You know, when you measure sprinters versus marathon runners, for example, is their genetic distribution of fast twitch, slow twitch a little bit different compared to the other ones, or is it a training question? I'm not.
Well, I just am more thinking about when like going back to aerobic versus anaerobic exercise.
Exactly.
It's
that's
like
exactly the point. Yeah.
Thank you for letting me monopolize this. This really helps.
Oh, this is okay. I mean, nobody else is talking in, which I really think this session is extremely useful because it helps me explain things, and every time you explain it a little different, you know. But then, yeah, so that's the fast twitch and the slow twitch. So that has to do with that aeration, the oxygenation, and the contraction. That's a continuous contraction uses much more of that ATP created with oxygen versus the ATP that we store in muscle. See, if you do a fast twitch, you're running a sprint, and then you're done with it, you can use up all the ATP you got, use up your glycogen reserves. You know, that's when we get into this creating phosphate, into this stuff, muscle metabolism. How are you making that muscle energy? How are you bringing it back? And the more longer term ATP you need, the more you rely on, or ongoing, not longer term, ongoing ATP you need for like a postural muscle, the more ATP you need from aerobic respiration, meaning you need oxygen to make the energy. You don't have stored energy. So in the muscle, we have a few ways to get this energy. We can get energy, and see in chemistry, if you go reading the biochemistry deep in the ATP, you start talking in a mitochondria how you make that energy. And you get into that citric acid Krebs cycle, RDA, blah, 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 which is just a bit overwhelming to go into too much detail. But we make ATP by eating glucose or by other stuff, and then that gets into this glucose pathway, and ATP comes out of it. And we can make some ATP without needing oxygen. But most ATP we make, we will need oxygen with it. Oxygen here, oxygen. See this, using oxygen, making ATP of a glucose molecule, if you go chemically, gives you, with using oxygen, gives you 36 ATP. Without using oxygen, gives you 2 ATP. So you have some ready right there to go in the muscle, but you're running out real fast. If you only can use make 2 ATP out of every little glucose you got. Or if you have glycogen storages in the muscle, right? The glucose store, you can chop off these glucose and make ATP, but only 2 of them out of every glucose. If you have oxygen present, you can make 36 out of everything, every one of them. That's a big difference. Gives you much more longevity. Ongoing ATP surplus, of course, or a supply, not surplus. And then in the muscle, we have what you said there, Nino, we have that creatine phosphate, which is just basically a molecule that is present that can give off the phosphate and bring it back to an ATP. You know, add a third phosphate from the ADP to the ATP and regenerate the ATP that way. But again, when you look at energy duration, that's why I put up that slide, is like 30, 60 seconds from here, 15 seconds from here, and everything else you want to go doing is going to be through this process. So if you're running marathons, if you run more than a sprint, you're going to rely on this process. So you rely on oxygen. That's where that, as you said, that's where the fatigue comes in. All right. Make sense? Yeah, good. All right, what else we got on this chapter? Anybody else? Anything else? So the rest of this chapter is basically where the muscles attached. They're attached to bones for the most part, the skeletal muscles. And then we got 2 places that we need to name those attachments so we can discuss them. We need to call it 2 different things so we can name each one of them. And we have one word that's called origin. That's where the muscle, so to speak, originates from. So that's where it anchors in, and that's that bony attachment. And then the other bony attachment is the one that has the muscle, the bone then move when that one contracts, and that's the insertion part of it. So those are the 2 terms we use there. And then we also have, of course, we described then a muscle action. What does the muscle do when it contracts? And that's the muscle action. So this won't be like flexing the elbow. And then when we bring that discussion in, then we just have a brief moment we can talk about and say like, well, one muscle contracts. That's fine, but how are we going to bring that elbow back down and lengthen it back and elongate it? What happens with that? Because the muscle only contracts, so it can only flex the elbow. It cannot extend the elbow. So then a discussion comes, well, we need a muscle on the backside as well that does that motion. So then we get from the biceps to the triceps back here. So we have one muscle that does one motion, the other muscle does the other motion. Depending on what the motion the joint needs to do, we're going to have all these muscles arranged in these positions geometrically that they can execute those motions well. And so when we look at terminology, we have the one that does the motion is known as the agonist or main mover. Where is that word? Do I have that? Agonist, I don't know if it's written down here. So it's called the main mover or the agonist. And then the one that does the opposite movement is down here, the antagonist, they call that. So you always have both of those muscles. You always have one and the other. What's interesting neurologically, if one contracts, the other one has to relax. Otherwise, there's no motion happening. And we can use that clinically later on if you got, when you do body work and stuff like that. So that concept, that neurological concept, we can utilize. And so and then we also have some muscles that help. Like if you only have one muscle that flexes the elbow, but we have some, side things going on and we're not quite straight in the middle, we need other muscles that help on the side to make that motion be executable. And those muscles are known as synergists. So when we then look at muscles, we look at all these different muscles with motions. And a lot of them help with motions. They might not be the main mover, but they help with motions. And then down here, lastly, we have there, or almost lastly, we have muscle shapes. Muscles have different shapes to them. And the big difference that I really think is kind of cool, we have this one shape, the fusiform shape, the parallel fusiform also. This one, conversion 2, those sort of have long cells that go from end to end of the muscle. So their contraction is a little bit weaker because we have other ones that have a long tendon and the muscle fibers are actually shorter and they go into that tendon and then their contraction level is shorter. So they have more muscle doing that motion. So they're stronger, those muscles. You can have multi of them. Pennate, they're called pennates. Pennate is a word referring to feather, because when you look at a feather, it kind of looks like that. That's kind of like a feather, or this kind of look. Unipennate, bipennate, even multipennate. So when we look at the strong muscles, we really look at these pennate muscles where they have the fibers go in an angle into a tendon that goes down the length of the muscle. So that's a little bit about muscle shape, just a bit more. That's like, going to be physics kind of thing, force transfer things. And then we have some helpers inside the muscles to help us smooth motions for the most part. So we have synovial tendon sheets or synovial bursa. Synovial is a fluid that's inside a membrane. It's kind of, you're thinking of a pillow. The feathery part is the synovial fluid, and then you have around that you have a membrane called the synovial membrane, and it has that space inside, and that's where that fluid is. And so it creates that pillow that prevents like muscles from rubbing over one another. For example, if you look at these here, one muscle has one motion, the other muscle has a little different motion, they start rubbing against each other. Rubbing does what? Creates heat. Heat creates inflammation. Inflammation creates pain. Oh, we don't want pain. So that's why we need these kind of things. So we have these synovial bursa that are between muscles, or we also have synovial tendon sheets that go around tendons. So that's pretty darn cool. So when these tendons move, it's kind of like cooling elements around these tendons. You can think of it like the cooling elements in the car kind of thing. This is like, yeah, that's actually not a bad way to look at it. See, that's how that looks from the side. So you have a tendon go around this. It's like a pillow that's wrapped around the tendon. So that's pretty darn cool. The other thing that I kind of think is pretty darn cool is, of course, we got fascia. Fascia is what connects everything with one another. So when you actually look at the tendons, really, at the end of the day, that's a fascial structure in some ways. Well, maybe anatomically, I got to be careful saying that. But from a clinical perspective, for sure, it works together like that. But the other thing that's really cool is the sesamoid bones. And the sesamoid bones are bones inside tendons, and they help the biomechanics of the muscle function motion. So when you look at this, when you look at the patella, when you have the kneecap, when you have the quads here, these muscles, big muscles here, which need to bring that knee up and straighten that leg out. Think about kicking a soccer ball. That's a lot of force. Your quads, if there is no patella, will go right from here, go straight down like that, straight like in a right angle, more or less. And so that force transfer is very weird, because that means you would have to contract the quadricep muscle very heavily until there's a response down here as the force goes straight down. But then all of a sudden, when there is a response and the knee will move, or the leg will move and straighten out, it will move like crazy fast. And so in order to smooth out the motion, to be able to have a continuous muscle contraction force and a smooth motion, we have these little bones that grow in the tendons, and they're kind of smooth motion makers kind of things, these sesamoid bones. So those are cool. We have some in the foot, some in the hand, all over the place, but that's kind of neat. All right, so that's that for that chapter. In terms of that, I wanted to go through that a little bit since there was questions, and I hope it helped you guys too. Is that so or not? How are you guys doing? Nobody.
I'm doing
Hello.
good. I understand everything.
Okay, good. Good. Was that helpful, or was that just review?
No, it was actually really helpful for me since I was taking down notes while you were speaking.
Good. So yeah, no, that's good. Yeah, those kind of chapters is not bad to do on Zoom. The hands-on's getting a little harder. So I think I don't want to keep you guys too much longer. Let's just, if you speak out, if have questions, just speak into it, interrupt me. Otherwise, I'll just want to go and talk briefly about this coming week. I mean, the second thing we did last week, or you're finishing up still, these weeks are a little bit fluid, are the trunk muscles. So we're getting into musculature of the trunk. For the most part, a lot of those are core muscles. A lot of those are postural muscles. And then this coming week, we're now going into the upper extremities and then the lower extremities. So we're going into the arm muscles. So there's only one chapter on this one. Well, not only, that's a lot of material. So go through that video stuff a couple times or once, and then do the quizzes or do that parallel. And then the labeling exercises, I want you to indicate muscle attachments as well. So you work together with the bullet points of the lecture material to fill out those, and that way you have sort of a routine of learning that. And then we have a discussion on the posture evaluation, although Nino, are you still here? I don't know if Nino's still here. I'm going to put this back 1 week. I'm going to put this back 1 week because we are making the posture evaluation 2 weeks. I'm going to put this back to the next week, actually. But what we're going to start this week, I want you guys to start with the injury prevention evaluation. And what I did there is I created a pamphlet for my patients, actually, originally, that I broad information from workshops and orthopedic literature stuff that are tests, orthopedic tests that are very simple to do, but give us a lot of information about the status of a situation. And so I want you guys to do that thing and answer some questions with that. So see, we have, like, for example, a one-legged test. This is balancing. This is not, well, it actually is related to the musculoskeletal system very heavily. And so then we have normative data that tell you, look, if you are this age, and you should be able to hold it so as long with your eyes open, with your eyes closed. And if you're not within that norm, I give you ideas of what to do to improve that. And all of those tests, like the squatting tests, how strong are your anti-gravity muscles, for example, or a side bridge, all of them are made, some of them are stretches, not just strengthening. So it's not just hard stuff, but they're made to identify things that often get hurt when we do, like, you know, weekend warrior type sports or not our elite athlete kind of people, because they are actually doing that as well, because this is the tests they use at the Swiss Olympics, people that test for them. And so I want you to go through those. You know, do not do them if it hurts. Don't push yourself too heavily, but it hopefully gives you an idea. There's like 10 of them or something like that. So let's see, there's a few of these things. Do the best you can with them and answer some of the questions. When I go into here, I have a questionnaire set up in behind. See here, there is questions. There's also a little video that I did, a 30-minute webinar kind of thing. And I want you to work your way through this. I give you guys 2 weeks for this one. Don't worry about, you know, doing it all at once. You can sort of spread it out a little bit. But that gives you sort of an integration reason of why we're learning all these muscles and what do they mean, you know, like, why are they important? We talk about those things. And so that's basically then it this week. And so that one this year, I'll move to the week 7. So don't worry about this. We'll do that next time. So you'll finish on the health kit side, you finish the ergonomic evaluation, do these exercises this week that I proposed in there, these little few things like neck retraction thing. They're good for anybody anyway. That, so, you know, prevention for when you're old, so you don't get old, is forever young. And then you start with the more active portion of the where was that? Injury prevention evaluation. All right, good. I think that's it. Any last questions, or can we close it out? Yeah, what a suggestive thing to say. I'm sure nobody has questions if I say it that way. Are you guys good?
Can you post this on YouTube? My phone was cutting in and out through the video.
Yeah, I'll put it up. I'll try to put it. Okay, good. No problem. Anybody else? No? Well, with that, I wish you guys a wonderful week. Be in touch. We're not in class today. I sent out some text in terms of in person. So no in person. I'll send out another note just now. That way, hopefully nobody will show up. But I will be there Wednesday. I just couldn't make it today, but I will be there Wednesday. So if you can make it, please show up. All right. With that, I wish you a wonderful week. Take care.
Bye. See you Wednesday.
See you Wednesday.